In cellular biology, a long-standing principle is that a protein's function is dictated by its three-dimensional shape. But what if some of the most essential proteins have no stable structure at all? Today, we're examining a computational tool named flDPnn, which is advancing our ability to predict and understand these structurally dynamic molecules known as intrinsically disordered proteins.
So, let's define what we're discussing. For decades, the paradigm was that a protein's function is encoded in a stable, folded structure. However, a large fraction of proteins contains regions that defy this model. These are intrinsically disordered regions, or IDRs. They exist not as a single shape, but as a flexible, constantly changing ensemble of structures. This presents a seeming contradiction. How can this lack of structure be essential for biological function?
These regions are not just functional, their proper regulation is critical for cellular health. When they malfunction, they are often implicated in the development of major human diseases, including various cancers and neurodegenerative conditions like Alzheimer's disease. This fact transforms the study of IDRs from a matter of basic scientific inquiry into a significant area of medical research.
In this explainer, we'll proceed as follows. First, we'll explore the paradox of these unstructured proteins. Next, we'll cover the computational challenge of predicting them. Then, we will introduce flDPnn and review its performance. We'll examine the architecture that enables this performance, see how it also predicts function, and conclude by considering the implications for future research.
Let's begin with Section 1, Proteins Without Structure. The central question here is how functional order can arise from structural disorder within the cell.
Here's an analogy to clarify the concept. A well-structured protein can be thought of as a specialized tool, like a wrench, designed for a single, specific task. An intrinsically disordered protein, by contrast, functions more like a multi-tool. Its flexibility allows it to adapt and bind to numerous molecular partners, which in turn allows it to participate in a wide range of cellular processes.
And it's important to understand that these are not a biological rarity. In fact, more than 30% of proteins in eukaryotes, including humans, contain these long disordered regions. Their high prevalence suggests that this structural flexibility is not an exception, but rather a fundamental operational principle in higher organisms.
Which leads us to Section 2, the prediction challenge. Given their importance, how do researchers identify these regions computationally? And just as critical, how is the accuracy of these predictions measured?
Because of the sheer number of IDRs, computational prediction is a necessity. To standardize the evaluation of these predictors, the scientific community established CAID. This is a blind assessment where different methods are tested against the same new data. It serves as the objective benchmark for performance in this field.
For some time, progress in this area had appeared to plateau. The best-performing predictors in CAID consistently scored below 0.78 on a key metric known as AUC, which stands for area under the curve. This metric quantifies how well a model can distinguish between disordered and ordered residues. Surpassing this 0.78 threshold became a significant objective for the field.
This brings us to our third section, which details the performance of flDPnn and its establishment of a new state-of-the-art.
This chart shows the result. In the most recent CID-TAD assessment, flDPnn achieved an AUC score of 0.814, clearly surpassing the previous performance ceiling. This represents a statistically significant improvement and sets a new benchmark for prediction accuracy.
The result is corroborated by other metrics as well. Using the Matthews Correlation Coefficient, or MCC, we see a similar outcome. flDPnn, on the left, scores 0.370, again outperforming the next best predictor. This consistency across different metrics indicates a robust improvement in performance.
Accuracy, however, is not the only relevant factor. Prediction speed is also critical for practical application. flDPnn can process a protein sequence in approximately 5 to 10 seconds. In contrast, other accurate methods can require several minutes for the same task. This efficiency makes flDPnn suitable for large-scale proteomic analyses, which involve processing thousands of proteins.
So, let's move to Section 4, how flDPnn works. What are the architectural features that enable this combination of accuracy and speed?
You might ask, what is the basis for this improved performance? It's not simply a matter of more data or more computational power. The advance stems primarily from innovations in the model's architecture.
We can break down the architecture into three main stages. First, it generates a sequence profile using evolutionary information.
The second, and key, stage involves encoding features at 3 distinct levels: the individual amino acid, a local window of residues, and the global protein level. Finally, these multi-level features are fed into an AI model, which generates the final prediction. The most notable innovation here is the inclusion of protein-level features. To quantify its impact, the developers performed an ablation analysis. They tested the model's performance with and without this specific component. As the chart shows, the full model's accuracy is substantially higher than the model lacking the protein-level features. The performance drop confirms that this global context is a critical element of the model's success.
Now we come to what may be the most functionally important development, Section 5, Predicting Disorder and Function. This is where the tool moves beyond simple identification to predicting the actual purpose of the disorder.
To put it simply, previous state-of-the-art predictors primarily answer the question of if a region is disordered. While useful, this is an incomplete picture. flDPn goes a step further by also predicting what that disordered region does. This shift from structural identification to functional annotation is a significant advance for the field.
The functional predictions are specific. The model is trained to distinguish whether a disordered region is likely involved in binding to other proteins, to DNA, or to RNA. It also predicts regions that serve as flexible linkers between structured domains of a protein. These represent 4 of the most common and important functions attributed to IDRs. This leads to our final section, the path forward. What are the potential impacts of this new predictive capability on biological research and medicine?
So to summarize, the advantages of flDPnn are threefold. It provides a new level of state-of-the-art accuracy. It does so at high speed, enabling large-scale analysis, and it offers an integrated prediction of both structure and function, which gives researchers a more complete biological context.
We are left with an important question for the future. We now have a computational method that can rapidly and accurately map not just the disordered regions within proteins, but also their likely functions. This capability offers a new level of resolution for studying complex biological systems. The question then becomes, how might this accelerated annotation of protein function advance our fundamental understanding of biology and, in turn, the development of new therapeutics?